Hello and welcome to Fast Medical Article Reviews. This is Mohammed. The article that we're going to be discussing today is called Intratympanic Diltiazem Chitosan Hydrogel as an Autoprotectant Against Cisplatin-Induced Ototoxicity in a Mouse Model. So basically what is this article about is that we're going to deliver this drug to the inner ear to the cochlea of the inner ear. And the drug is diltiazem, which is a calcium channel blocker that may have shown that it's gonna protect the inner hair cells from apoptosis caused by some toxins the chemotherapy that's caused by the chemotherapy cisplatin. And this drug is gonna be embedded in this CGB hydrogel, chitosan hydrogel, and this hydrogel has been described as a vehicle for drug delivery that can provide controlled, sustained delivery of drugs into the inner ear. And as the title says, it's intratympanic. The drug is going to be delivered intratympanic wise. So it's going to go through here, and it's basically it's going to be injected. This hydrogel medicine complex is going to be injected through the intratympanic membrane in the middle ear, and then it's going to diffuse through the round in the membrane and reach the cochlea and the hair cells. So this slide is about quantification of cochlear hair cells. You know, we want to see if the hair cells, not if the hair cell numbers are gonna be higher if in case they were protected by the diltiazem drug. If they were the same as the control, then they were not protected. But if the number was higher, then it tells that the drug has actually protected it. So, the outer hair cells are these three rows of hair cells, and the inner hair cells are this one row here. And so these histologic sections, you can see this column, it talks about the diltiazem, which is a drug of interest. And this column right here talks about saline, which is the control. This row over here talks about the apex. By the way, so these two figures, this one is about the inner hair cells number, and this is about the outer hair cells number. Orange columns are the number of viable cells after the control group, after the control drug, after the control is administered, and the blue columns is about the hair cell number after the drug diltiazem has been administered. This is the apex. So this row is about the apex, and this row is about the middle part of the cochlea, which are these two, and this is about the basal part of the cochlea, which is about these two. So we're gonna now talk about the only statistical significance found in this slides. On these slides, you see the statistical significance over here. So as you know, the number of hair cells in the drug groups are supposed to be higher than in the control group because they were protected. But actually, surprisingly, we see they are less in number and they are damaged. And as you can see, the column of the drug is shorter than the column of the control, which means the number of inner hair cells in the base was lower. And as I've already mentioned, the statistical significance is found only in the base and the inner hair cells, not the outer hair cells. So it's in the favor for the control group. And anyway, this is likely to be physiologically insignificant. This slide is about the quantification of ribbon synapses in inner hair cells of the apex. So ribbon synapses, they function to connect inner hair cells to spiral ganglion neurons. So they're important for the function of the inner hair cells and for the hearing process. And we want to see if the medication diltiazem has actually protected these synapses from destruction caused by the cisplatin chemotherapy. So the green color here represent inner hair cells of the apex, and the red dots represent the ribbon synapses. This column again is it's for the drug group, and this column is about the control group. Um, and this figure illustrates the number of synapses in the inner hair cells in the apex, middle, and base. But again, these slides are about the apex and they represent this column. And we brought these slides only in the apex because they were the site of the only statistical significant, as you can see here, of protecting the ribbon synapses. So as you can see here, the blue is the drug group. It um, and the number of synapses was significantly higher in the drug group over the control group. So we can conclude that there were significant protection by diltiazem from the cisplatin-induced loss of ribbon synapses. The change in the middle and the basal part of the cochlea weren't significant. And one hypothesis that can explain this is that a previous study showed that intratympanic therapy can penetrate the otic capsule and cause higher drug concentration in the cochlear apex by filling the bulla. Thank you for watching. I'm gonna leave a link for this article in the description, and thank you. Bye bye.